So I need to introduce Dr. Shiv. Shiv has already been introduced yesterday, so I will not go into details of his introduction again. But just to say that he gave a very nice talk yesterday. Professor Shiv Pillai is professor of medicine and health sciences and technology at Harvard Medical School, Boston, USA, and very kindly agreed to give two talks instead of one. I wrote to him, can you give me one talk? He said, I can give you more if you have time. So it is a privilege to have Shiv again and give the stage to him to talk about COVID-19 and his lab's experience or immunology about COVID-19. Shiv,
So I'd like to thank Narinder for a very nice talk, and again, Sunil and Amit and others for the invitation. I am going to say some points on which I may not have a complete agreement with Narinder and many on which I will. So let me start by saying that many of the studies that we are doing, we have to recognize what a terrible toll this disease has taken in disrupting lives, in affecting people who have, suffered from the disease and also from the mortality of this disease. So I'm going to start with a little homage to them, and then I'll get into the talk. So immune cells, they tell their stories through signaling cascades. Their tales are never one-sided. A fine balance pervades. What goes up, it has been said, must someday come down. Moderation is the key. There's no need to go to town. Our species, it was naked, faced a foe it had never seen. No antibodies in any human to the COVID spike protein. An immune system that is balanced, that generally is the norm. But sometimes immunity goes berserk and the virus starts a storm. Remember William Butler Yeats, but things don't always fall apart. It's when that fine balance is lost that signals go off the chart. Uncontrolled gene expression or a lack of inhibition, and the immune system loses its exquisite precision. That balance, it is crucial. Homeostasis, if you please. The loss of immune precision is the basis of disease. Yeats said things fall apart. The center cannot hold. Unregulated COVID immunity has sadly culled the old. So let me share my screen now and talk about the science. I hope you can see my screen. So I'm actually going to give you a little bit of background, which will go, into some directions that Narinder may not have covered. And I'll give you my opinion on what I think is going on, and then talk about some of our recent data that was published. We have some more things that are interesting that are going on, which I may not have time to get into, but I can discuss in your questions. So the first thing I want to emphasize that I actually think this decline in antibodies is not something that I would assume is commonplace, because if you get measles, just one measles infection, 80 years later, you will have very good antibodies. Okay, this we know. And this is true of many other viral infections, where you make antibodies, the titers actually rise, and then they stay up for your life to a reasonably high level. In the case of SARS and MERS, if you go back and look carefully at the SARS and MERS data, so I'm talking about what Narinder covered, 2002 and 2012 diseases, for most people, a small number, and I'll try to explain that later, a small number had antibodies 10, 12 years later, very small number. The majority of the people who survived did not have antibodies beyond a year. Okay, so it declined and kept declining and then was gone. So in COVID-19, we do have class switched antibodies. They are neutralizing. As you know, in many people they decline, and some they do persist. But what we also have noticed from the very beginning, from the first data came out from China, we looked at the sequences that they released at that time, and there's very little somatic hypermutation. So this suggests to us that in most people who get infected, whether it's mild, moderate, or severe, in most of them, true memory may not be generated. I mean, just finding a CD27 positive B cell doesn't mean that's true memory. That might fade. So why is this? This is the question we wanted to ask, among many other questions we're interested in. And as I emphasized to you yesterday, I said that if you're going to study a disease, you have to look at the blood. So we look a lot at the blood. And so some of the data coming out of blood, I don't agree with because it doesn't match in other tissues. So we look at blood, we looked at lymph nodes, we look at draining lymph nodes. So in all the severe patients that we could get immediate autopsy material on, and some of this was done by others on single cell RNA-seq, but the approaches we used have worked better, as you will see. We looked at draining lymph nodes, and we looked at the organ that is damaged. And we looked early in disease, when they died early, unfortunately, and we looked late. Okay. So just to recap, you all know that the RBD is the stands up in order to be bound to ACE2. So RBD is actually, this domain has to stand up. It comes in two different conformations. And if you looked at the D614G that Narendra talked about, D614G stands up much faster. And this is Jeremy Luban's work. He's somebody we talk to a lot. And when these stand up faster, of course you can bind, but what really improves is also the fusion event, because you bind and then you get cleaved pretty well, and then this allows entry. Okay? The affinity does not go up. The other thing that we want to consider in disease, so of course, type 2 pneumocytes will get infected first in the lung. It all starts in the nose to begin with, of course. But remember that once, of course, the ACE2 binds to RBD, in most cells, TMPRSS2, the infected cells, we know they're readily infected, TMPRSS2 will cleave off the head. So all this RBD part and all will be gone. You can think of a spring-loaded clip underneath, which is this fusion domain, which will allow this membrane, so this protein will insert itself into the host membrane, and then it's a calcium-dependent membrane, it will spread out, and this viral membrane will fuse with the host membrane. Now, unfortunately, this virus has a particular positively charged sequence in the spike, which allows cleavage by furin, and furin is widely expressed. So late in disease, when we see viral spread and then you're getting into many other cells, possibly, we know of many examples, this might be due to furin. So that this is another problem with this virus. One is easy entry. It's transmissibility is high, but also it can enter a little promiscuously. Okay, so this you all know. Well, the reason why we all have this severe disease and why it might ameliorate a little bit with time, but that's also a little doubtful, is that there are no pre-existing antibodies, not even cross-reactive antibodies. This, this RBD is fairly unique. So we don't have anything from a previous common cold coronavirus or something else, which helps us a little bit. So unless you have neutralization, you're going to enter the cell. Antibodies are really important in the first few days of infection, because that's when the virus is spreading. After that, if you've got a lot of infected cells, antibodies do nothing for you. Okay, what you need is antibodies early on or before you're infected. So my worry is, of course, if antibody levels decline, you may create a little bit of memory later in infection, but you didn't have much antibody made because antigen got lost in this disease. You may not be protected from reinfection. Okay, because it depends. On a small percentage of people may have good enough antibodies and memory cells, but just having memory cells doesn't protect you from reinfection. It says that you will start making antibodies fairly soon after getting infected, and hopefully you don't get very sick. But maybe you will get sick. There's nothing very clear about that. Okay? All right. One of the other things about this virus, and this is true of some other viruses too, is that it evades sensors. So when you think of how do you make type 1 interferon against an RNA virus, this is a positive strand RNA virus, short double-stranded viral RNA can bind to RIGI. You need the 5' end to be free. It has to be just 5' triphosphate. And longer viral RNAs, double-stranded RNA, so when you replicate, these can bind to MDA5, but in this case, MDA5 requires that not only should you, it doesn't mind if you have a cap, it doesn't mind if you covered up that 5' triphosphate, as long as you don't have an extra modification, which is this 2' O methylation of ribose, the first ribose after the cap, then you're going to allow the sensor to respond. So our messenger RNAs not only have a cap, which allows better translation, but they also have a second modification precisely made to distinguish us from viral RNAs. Okay. The other thing that you probably know is these interesting papers showing that these coronaviruses, they replicate in double membrane vesicles, which bud out from the ER. There are a couple of proteins of this virus which allow the formation of the double membrane vesicle. The virus is replicating in a hidden way. So it's hiding from, most of it can hide from MDF5 and from regae. There's a very interestingly also a pore made by one of these, a beautiful structure here. This is like a work of beauty, this protein. This is the, one of the non-structural proteins of the virus, NSP3. It makes a pore from which the RNA can come out in order to make more virus and get out of the cell. Okay. The point I'm trying to emphasize is a lot of the proteins in this virus are designed to prevent the induction of the antiviral state, which I talked about yesterday. And these proteins, if you look at this, so normally on a messenger RNA, other than the poly A tail, we modify this 5' triphosphate by putting a 7-methyl guanosine cap. Okay. We also add this other thing, what I call the tassel after the cap. We also add the 2' O methyl group on the first ribose that follows. This virus has all the enzymes needed to put a poly A tail, to put the cap, to put this methyl group, and to put this methyl group on the second ribose as well. So it is 1/3 of its genome in terms of coding material is designed to evade innate immunity and block the antiviral state. This is its potency. Okay. And so if you think of what is happening in a moderate illness or a mild illness, or even maybe in an asymptomatic person, that the virus gets into the nose, okay, typically. In the nose, let's say it got into this cell, it started replicating. The virus has the ability to prevent the cell from making type 1 interferon. So it's not going to make type 1 interferon from this cell to generate the antiviral state. But at this stage of infection, we have plenty of PDCs, okay, plasmacytoid dendritic cells. We have some macrophages, plenty of macrophages in the lung. The PDCs are the best cells to make type 1 interferon. So now they're using TLR7, a little bit of TLR3, but mainly TLR7, and they're using it to start to make type 1 interferon. This type 1 interferon will bind to type 1 interferon receptors on all the other epithelial cells, induce the antiviral state. So we discussed this briefly yesterday. And this induction of the antiviral state protects you. And for many people, this may clear the infection. In many people, all the asymptomatic people, this just happens automatically and you don't know about it. It's done. The antiviral state is favored heavily over inflammation here, because if you got rid of the virus, you're not going to get inflammation. And this is the best way to control the virus. Now, because we talked a little about T cells, I'm going to say theoretically, and there is no question that if you have a mild, very mild response, you don't get much of a T cell response because there's not much antigen. You go a little worse, not very sick still, you will get a T cell response because there is antigen, right? There's some more virus. But does that mean anything? Is it protective? Are those CD4 CTLs that Pandurang Anandan group found, are they protective or are they causing disease? These are questions we should ask. Okay? So I'm going to say that, yeah, probably, because type 1 interferons can activate. So not only do you get, protection by antiviral state, you'd activate NK cells. So early in infection, NK cells should be more active. They should also help you clear the virus. Early infection, you will also, cause type 1 interferons to cause better activation of lymphocytes on the lymph node. We know that, you'll induce CD69 and so on. Your T cells will multiply more. So you could make cytotoxic CD8s, for instance. These cells could be made in the draining lymph node. So this would be, the retropharyngeal lymph nodes and other lymph nodes that drain the nose. And then your activated CD8 cell could come and kill virally infected cells. This is theoretically possible. Whether it's even necessary is unclear, because you might be controlling things by just the type 1 interferon at that early point. So this, Narendra referred to this, 4% of people have mutations in the type 1 interferon TLR3 pathway, and about 10% of people have autoantibodies, pre-existing autoantibodies against type 1 interferon. So if you're asking scientifically, why did some people get very sick? Yes, one possibility is that they had some defect in this type 1 interferon pathway. So they couldn't protect themselves as well early on, and this happened. Why do older people get this more? I don't know. So the concept that Narendra brought, it's a very important concept about India and Africa, and we always think about this. Maybe trained immunity, the idea that Mihir Netea has put forward, it's a variant, a major variant of the idea that is coming from the idea of hygiene hypothesis. So trained immunity, and they have a nice paper out in Cell recently showing that BCG vaccination is protecting people from viral infection. So it's possible BCG, other infections are protective in some way, but then there are still susceptibilities in people. And those susceptibilities probably relate to a failure of the protective part of innate immunity, which then allows the inflammatory part of innate immunity to take over. Okay, and that's the real problem in my view of this disease. Most of the problem is coming from defective innate immunity or too much of innate immunity of the wrong type. Okay. What is the role of T cells? We should think about it. So you think about the virus and you say, okay, here's the virus. Of course, RNA is a PAMP. That's possible. N protein has been claimed to be a PAMP. The data is a little weaker on it. There's a preprint. We don't know whether it's real or not. Spike has been shown to be a PAMP. Spike is actually pretty good at it is very potent at activating the alternative pathway of complement. So nice work from the Brodsky lab on this. So I think this is a major thing. Death, damage, DAMPs being released. These are also driving inflammation. So what you see early in disease is severe inflammation with neutrophils and macrophages. And we have looked extensively in the lung. So we published the lymph node data and the blood data. We didn't have it yet published the lung data, but there aren't many T cells in the lung early in disease. Even in severe disease, it's hard to find the T cells there. You find, we quantify everything, and I'll show you some of the data now. All right, so at the end of the day, you get inflammation, you get cytokine storm, and much of the cytokine storm does not require to be T cell dependent, okay? The evidence that is driven by T cells is not that strong, at least early in disease. Later in disease, there may be some role for T cells. If you survive for 2 months and then you're still very sick, that might be the case. Okay? So why do viral PAMPs not trigger antiviral state in severe COVID-19 through PDCs? Okay, this is an important point to know. And I think you need to understand that in any infection, any severe infection, okay, there's a block in thymic output and there's a block in the bone marrow. And the way I would try to look at this is that if you divide all your cells, and I'm showing you some of our data here, one thing we know for us, PDCs, we showed, and also Ginho's lab, Florent Ginho showed that PDCs are actually lymphoid-like cells. We showed that they're unique progenitors that make PDCs. So lymphoid-like populations and lymphocytes diminish dramatically in severe disease. So PDCs go down, NK cells, this starts to happen fairly early. NK cells start to go down, they go down dramatically. Although NK cells are one of the few cells which increase with age, okay, they actually decrease dramatically in COVID-19. And I'm talking now, in general, blood, lymph nodes, and in the lung as well. ILCs, B cells, T cells, all decrease. Okay, this is pretty dramatic. What goes up is myeloid cells, macrophages, monocytes, and so on. Now, why do I say this might be a bone marrow block? So in our work, and I'll come to this work in great detail towards the latter part of this talk, when we look at the earliest B cells, transitional T1 and T2 cells, and we look in severe patients, as is shown over here, the T1 and T2 cells are lost. Okay, so what was happening in COVID for us, what we see is the earliest cells that come out of the marrow are already lost in severe disease. And they're declining, by the way, even as we monitor from moderate disease to severe disease and convalescence, these decline. So what's driving this? Part of the thing we cannot forget about is complement. There is evidence all over the tissues for complement activation. And so we have a few things to worry about. We also have blood clots forming in every organ and hypoxia being generated by that. So There is good evidence for how you can activate the alternative pathway of complement, and that spike does that. There's a lot of evidence for netosis. You'll find nets everywhere. So in the tissue, viral infection could cause some damage, but don't necessarily always have viral infection on the endothelium. This causes release of tissue factor. This is going to allow between complement action, C5a will activate your nets as well, your neutrophils, and form nets. And remember, when you activate complement, you can activate coagulation as well, and vice versa. And you have a lot of coagulation factors being made in very large amounts because you're making so much IL-6 and IL-1 and TNF and so on that's going to drive the liver to make a lot of fibrinogen. So it's a very easy thing to find. C-reactive protein is a good measure of disease severity. So are D-dimers, which are fragments of fibrinogen. So I think we have to think along these terms, and then we can start to think about B cells and T cells and everything else. Okay, the other background I want to give you is, when you think about germinal center formation, so yesterday you got some of this from Joshi. When you have an initial TB interaction, you form something called the extrafollicular focus. The T follicular helper cells here are the kind of cells that are being seen in the blood as well, by the way. You cannot find real T follicular helper cells in the blood. They do not exist in the blood. Anyone who thinks they are really T follicular helper cells with BCL6, that is not true. What they are looking at is what we call pre-germinal center TFH or extrafollicular TFH-like cells, okay? These are the cells which we showed a few years ago are the drivers of class switching. We showed that in humans. That's also been shown in the mouse more recently. So it is here that you get the action of forming weird activated B cells in disease. Okay? The germinal center we discussed, of course, is key in order to get high affinity antibodies, to get true memory B cells, and long-lived plasma cells. I don't think we have still got evidence that, except in a small proportion of people, and I think there's nice data from Patrick Wilson, small proportion of people who survive after severe disease, they might be making some long-lived plasma cells and long-lived antibodies, but it's a very small minority. Now, if you look at the germinal center, this is some staining from our lab. AID is the enzyme that drives somatic hypermutation in the dark zone. This is how you mark the dark zone. ICOS is a protein on the surface of T follicular helper cells. Okay, and ICOS is, so T follicular helper cells are in the light zone. BCL6 is the transcription factor both for germinal center B cells and for true T follicular helper cells. This is the germinal center. This is all from a normal thoracic lymph node. Okay? So we know in the dark zone of a germinal center, you have somatic hypermutation, then you have selection, rounds of selection, and the high affinity cells. First round, you'll get some memory. Later on, when you get even higher affinity, you become long-lived plasma cells. Okay, this is what we understand about the germinal center reaction. So our thinking was, okay, SARS, MERS, common cold coronaviruses, you get reinfected multiple times in the same year. Why is that? Why is this virus so, the same virus? It's not like some new strain. It's the same virus that's infecting you again. So we started to wonder, what is going on with these viruses? Why do you have that happening? And this is what led us to some of our lymph node studies. So again, to re-emphasize, if you think about T follicular helper cells, there's a stage where we call them pre-GCTFH, which I'll describe to you. And pre-GCTFH are driving class switching, and they are relevant in disease. I think in many ways, we should think about them. But they are outside the germinal center. Okay, GCTFH are in the germinal center. They have a high level of BCL6. Okay, this is what I just want you to remember. So now when we looked at lymph nodes from patients, so this is we're looking in a control. These are all thoracic lymph nodes. And these are age-matched individuals who died. This is all from the Brigham and Women's Hospital, where Amit used to be at one time. And these age-matched patients who died of some other cause, and it's not COVID. Okay? So these are our controls. You can see nice BCL6 staining. These are the germinal centers that you can see. And if you look in a patient, this is late COVID. I actually think in late COVID, you lost your germinal center. That's trying to come back. They're not yet come back. They will, if you are convalescent and you survive, you may get some back, but they are, your virus may be gone by then. This may be to something else, but you may get a little bit of response then. So if you just look at BCL6 now, just look at the control first. Beautiful germinal center with BCL6. Look in early disease. These are people who died within 8 days of the initial onset of symptoms. Okay, and this all was happening in April and May in Boston. So this is BCL6, the very poor formation of attempt to form germinal center. Here's in late COVID, you see some more cells, they're coming together, but when we quantitate, you won't see much, as I'll show you. And of course, controls are good. What we found interesting was AID. AID is expressed really nicely and tightly because you've got a nice germinal center here. But there is plenty of AID in both early and late COVID-19. So AID cannot be induced until the T cell activates a B cell. So this TB collaboration is what we inferred at this point, and we wanted to test that. And AID was not defective. Okay. This quantitation. Total number of B cells. This is, we're looking again in thoracic lymph nodes. Total number of B cells are down. Okay. And we're early, later in different colors. Total number of T cells is down, even in the lymph node. Total number of germinal center B cells. This is again, remember we are doing it in two ways here. One in this way is the number per square millimeter. These are absolute numbers, not just some percentage which you get from flow, which is sometimes can be deceptive. This is absolute numbers. This has gone down dramatically, BCL6 positive. Now, if we look at a proportion of CD19 positive cells, still down. Okay. AID positivity down in absolute numbers. Remember, there is lymphopenia. But you look at proportion, it's not that bad. Okay, this is in the lymph node. And again, I say that because AID seems to be somewhat preserved. Here is in the spleen. Now, remember, there's viremia, and in some people, clearly the spleen is going to be affected. And when you look at the spleen, again, we're looking for germinal centers. Again, we're looking in controls and patients. And what you can see here is, okay, you can see some germinal centers in the controls. Here's a beautiful BCL6 positive germinal center in the control. Patients, they're lost. We quantitate total B cells are down in the spleen. Total T cells are down in the spleen. Quantitate BCL6 positive cells down in the spleen in absolute numbers, down in proportion somewhat. AID positivity is down in absolute numbers, proportion-wise not so bad. Okay, so again, the big picture message is, yeah, you in the germinal center, you're trying to make your germinal center response, you're failing in COVID-19, but you're still activating, getting TB collaboration. Okay, this is looking at T follicular helper cells. I'll just touch on one or two aspects of it. Again, so the hypothesis in our head was, maybe you can't make the T follicular helper cell, and if you can't make T follicular helper cell, you won't get a germinal center. This is BCL6 staining in the control. This is BCL staining in a late lymph node, BCL staining in early lymph node, okay? And we are looking for CD4 positive, CXCR5 positive, ICOS positive cells, and we're looking for BCL6, okay? These are the things we want to look for. When you do quantitation over here, you'll notice that when you look for CD4 positive, ICOS positive cells, this early and late, it's down significantly in COVID-19. If we now look in for CD4 ICOS positive as a percentage, again down, this better now. This is not that you lost all your TFH cells completely, but you lost the BCL6 positive guys. BCL6 positive cells down significantly in numbers. BCL6 positive as a proportion, it's down. Okay, so that was all lymph node. This is in spleen, broadly similar phenomenon, okay, that you're losing BCL6 positive cells and TFH cells. What about all the other T cell subsets? So I'm sorry, Amit, we didn't look at TH9, but we looked at TH1, TH2, TH17, and Tregs. And you can just gauge by, I'm going to show you the staining for different transcription factors. This is TBET. So this CD4 positive, TBET positive. This is GATA3. So first thing you can see is there's some increase in TH1s. This is all as a proportion, okay? We'll come to numbers and percentages in a bit. You can see an increase in proportion again, a decrease in proportion of TH2. This is control, and you have reduced TH2 in the disease. TH17, slight increase, okay, in the disease. Proportion only, not absolute numbers. Tregs, proportion-wise, early less, late in disease, there's some attempt at recovery here, a lot of Tregs now, okay, as a proportion. This is the control. So now if you go through quantitation again, if you're looking at, this is percentages, all percentages over here, you have more TH1s, less TH2s, some increase in TH17, and Tregs change. Okay. We did quantitation also, it's all in the paper. I'm just going over the data briefly. We looked, because we can look at cell-cell interactions and measure true cell-cell interaction by multispectral imaging, we're able to show lots of TB conjugates. This is an example of TB conjugates. We also found a lot of cells in the lymph node, which are IgD negative, CD27 negative. We call this population of IgD negative, CD27 negative cells double negatives in the B cell field. And there's a lot of subtleties to it. And I think these are all the B cells that have been variously called age-related, or sometimes they've been called exhausted by different diseases. But in reality, they are very much disease-related B cells. They might cause disease, that's all still speculative, but you see a lot of them in this disease. So then we also knew from the literature that there are a few papers that have been published over the years where you lose germinal centers in the mouse. I mean, everything mechanistic we learned from the mouse. And we knew in a severe malaria model, you lose germinal center cells, you lose TFH cells. And then when they blocked TNF or they blocked interferon gamma, these came back. There's another model of Ehrlichia muris, which is a rickettsia. In this model also, you lost germinal centers. Okay, and again, TNF blockade, not just knockout, TNF blockade reversed it. Then Rafi and Dan Baruch had published a paper, and we have recreated this model in the lab, where they didn't look at TFH, but they found loss of germinal centers. They were trying to do a vaccination, and it turned out to go in the wrong way. The mice all died. Okay, this is a strange story. I can tell you more about it. But it's a beautiful model to study this phenomenon. And then they have severe lymphopenia. It looks like they got SARS-CoV-2, but they gave them LCMV. Okay, but it's a pre-immunization. So there are models which tell us, okay, you can study this more, and this is what we do now. So what this led us to think, is TNF the cause of this problem? Maybe it is. We can't prove anything humans. We can only correlate it. So we went and looked. So this is what you normally see in a lymph node. Okay, this is a control lymph node, again from old fellow. The control lymph nodes always have few TNF positive cells, mainly in the follicle area. In the disease, TNF alpha is all over the place in the lymph node. The lymph node is such an interesting thing to be studying this disease, but this milieu has changed. A lot of TNF alpha, some of it macrophages, some of it other cells. If you quantitate and you look at TNF alpha cells by quantitation per square millimeter, they're increased quite significantly in COVID-19. This is, total percentage, again, increased in COVID-19. So the model we came out at this point, and I'm going to come to the blood in a minute. The model was that, okay, in normal, if you got measles, you'll make some TH1, TH2, TH17, maybe some TH1, hopefully. You make some activated CD4 T cells. You got TB collaboration, and B cells help T cells. They make TFH. The TFH differentiate into BCL6 positive TFH. This is the way things should happen. And the differentiated B cells, the TFH cells, then go in and make the germinal center. And that's why you get antibodies for life. In SARS-CoV-2, both in the spleen and in the lymph node, first of all, severe lymphopenia. Secondly, proportionately high increase in the lymph node of TH1, increase in TNF secreting macrophages and other cells, lots of TNF. And we see a block. We see all these dysregulated B cells, the double negative B cells. And we see a block in TFH differentiation and no germinal centers. Okay, so this is the big picture view from looking at the lymph node. Look in the blood. So we typically do these 25 color, 24 color panels for T and B cells. So actually we have a lot of data on CD4 CTLs also, but I'll tell you, because of what we have looked in the tissues, we don't think it's as important as maybe thought, okay, on CD4 CTLs. Anyway, so we have 25 color flow cytometry. We have B cell myeloid panel and so on, okay. I'll just give you a general view of it. So looking for plasmablasts, here's control. Moderate disease, you can see an increase in plasmablast. Severe disease, you can see actually proportionately, just look at the other cells here, very high increase in plasmablast. During convalescence, you're coming back towards equilibrium, okay? Let's go and look at follicular cells. These are normal follicular cells. Start to decrease in moderate disease, gone in severe disease, pretty much reduced, and then they come back in convalescence. Let's look at those early transitional cells. Transitional cells, they're in controls, start to go in moderate disease, gone in severe disease, come back in convalescence. Now, again, look at the proportion of the double negatives here to everything else. Double negatives increase in proportion, become the dominant cells late in disease, in severe disease, and then start to come back to equilibrium after convalescence. Okay, and these double negative cells, I should tell you, are the ones we are seeing in the lymph node outside the germinal center, and they are coming out into the blood, and we're looking at them. Okay? This is just telling you we did a lot of clinical correlation. So Jocelyn Farmer is in my lab, who is one of the clinical immunologists. So she, looked at all the records. We did everything to measure this, and basically, as disease gets worse, transitional follicular cells decrease. Okay, predictable. As disease gets worse in different measures, so now we are looking at CRP levels, and we're looking at naive cells, follicular cells, transitional cells. You see the declines. Then we see symptom duration at the time of blood draw. How long do they have symptoms? You see decline that goes along that line. You see total hospitalization stay. And again, you see there's a decline which goes with severity. Okay. Okay, then what about activated disease-related B cells? So there are many categories. I don't want to, bore you with the kind of minuscule details we like to look at. So we have many subcategories of activated cells. We're learning so much more in the last 5 or 6 years about novel human B cell populations. But let me just walk you through, say, that if you thought about all the weird B cells that we see, they increase with disease severity. And if you look at this chart at the end and say, okay, how do we look at activated B cells totally as disease severity goes up? You can look at this going up, or you can look here to see the proportions of all the cells going up with disease severity. So as disease severity grows, plasmablasts, activated B cells, switched memory cells, which we do not consider to be true memory because we're calling them switched memory because they're IgD negative, CD27 positive, and they're all increased. Okay. We also looked at absolute numbers. So because we took fresh, this is not done flow after freezing. This is all flow done fresh from every patient. So a lot of people put an effort into this. We could look at absolute numbers, and the same phenomena hold true, that the weird cells go up in absolute numbers, and you can correlate this with CRP, you correlate with symptom duration. Then what about, are these cells antigen specific? So what we did was, so obviously the RBD positive cells are going to be a small proportion of all antiviral cells. There's a good measure of specificity for SARS-CoV-2 because this RBD is not shared with anyone else. So we use 2 floors. So we're labeling RBD with PE and APC, and then we're gating in convalescence or in severe disease, for instance. We're looking at RBD positive cells, and then we're going by flow and seeing what all the populations were seen. And this is just a measure patient by patient. What did we see in terms of RBD positive cells? And of course, they're not very different, whether you're looking severe disease or convalescence or the few moderates we looked at, the proportions are very similar in RBD positivity. So the weird B cells we're seeing in the extra germinal center area in patients in the lymph node and in the blood, what we see in severe disease, we are seeing even in moderate disease, which makes us think maybe the germinal center is not that great even earlier on. Okay, this is just quantitation. So the big picture view, in many severe viral infections, and you can go to Ebola and Marburg and everything else, you get cytokine storm, you get T lymphopenia, you get non-durable humoral immune responses, best studied in SARS and MERS. In severe COVID-19, we're seeing this decline in early B cells. So let's worry about T1, T2, and follicular here. We see an increased in activated B cell populations. We see shrinkage of the white pulp. We see germinal center collapse, preservation of AID positive cells, but loss of T follicular helper cells. Okay. So again, just to give you a sense, this is what happens normally, right? You get formation of germinal centers. In disease, you get activation of T cells, but lymphopenia. Okay, remember everything is in the context of severe lymphopenia. You get proportional increase in Th1s, but even with lymphopenia. You get TB interaction. It's not that T cells didn't talk to B cells and kiss them. They're inducing AID. They're making these double negatives. But then you can't go further. You can't make the TFH cells of the best type, which gives you the real high quality humoral immune response, which will give you true memory, and which will give you really good long-lived plasma cells. So I do think, and I'm talking to people like Patrick Wilson, I do think that people who get severe disease go into convalescence, okay, and they're now in the convalescent state. Uh, if they have recovered, if they didn't die, yes, some of them will recover if they were lucky to preserve a little bit of the virus still, which unfortunately is generally not the case. The virus goes away after 1 week. By 10 days in most people, there is no virus to be found. Uh, but if the virus persisted when they could make germinal centers, then they might make true memory and they might make long-lived responses. But from my perspective, to hope for herd immunity from infection, and Narendra agreed completely with this, is not something that you should hope for. Okay. First of all, not only will you, even if you infected half the population, only a small fraction of them are making good antibodies. So where is the herd immunity going to come from? You're going to let half the population get infected, and then only one small fraction of it will be protected. There is no chance of herd immunity from that, if you believe antibodies are useful. T cells, there's no question T cells get activated. But where is the data to say they're protecting us? We have already shown, so 2 papers from Shane Crotty in Cell and 1 in Science, showing that we make CD4 responses that are cross-reactive with common cold coronaviruses. Sure, we make those responses. They didn't protect us. So I don't know what the protection is coming from. In India, I think it's something different. It's not common cold coronavirus that's protecting, it's other things. Could be BCG, could be other infections. These are all, is maybe attenuating disease, maybe allowing some better response earlier on, but we don't know that mechanism yet. But I think to hope, of course, there will be T cell immunity. And I would love to have T cell immunity protecting us. But whether it actually protects, there is actually no evidence. Whether T cells actually cause the disease, again, there's limited evidence. I would say early in disease, there's between us and what has been done by David Ting and others at MGH, we don't find T cells early in disease. And we've had the opportunity to go and look, in people who unfortunately died of some other cause. We have a range of patients from whom we've looked at autopsy. So even in them, we can't find many T cells. So I would be hesitant to say that T cells are driving the cytokine storm. They could late in disease contribute to the cytokine storm. But the main problem seems to be imbalance of innate immunity. So this is the big picture. What do we think happens in severe disease? Early infection was not controlled for whatever reason. We don't know what the underlying causes all are. There's some clues coming from the type 1 interferon studies, which are helpful. There's various ways the virus is subverting us. We get immune activation in draining thoracic lymph nodes, a lot of immune activation. I should add a few points to you. I actually don't think there is any evidence, true evidence for exhaustion. In fact, there's hardly any CD8 T cells going to the lung. I do think there is evidence, and we will publish this hopefully soon, showing you that there's huge activation induced cell death in the lymph nodes of CD8 T cells. So this virus is causing lymphopenia, like many severe infections do. It is depleting CD8 T cells dramatically. Even there's lymphopenia, and then in the lymph node, it's causing activation induced cell death of your CD8s. There's germinal center collapse. Okay, this we have evidence for. So it is able to subvert adaptive immunity as well because it has persisted and is in us for a long time. Then you get all the activation. Maybe lymphocytes activate macrophages and neutrophils in the lung. I think this happens late. But clearly early on, we have a lot of activation. You could have complement turning on the entire, innate immune system in the wrong way and activating neutrophils, macrophages, and so on, which might be the real problem that we have. All right, so this is the big picture view. I should thank a few people. So I mainly emphasize one paper. We have 3 other stories that I haven't got time to get into. These people, Naoki Kaneko did all the tissue work. Xiaoshuan Ko, Julie Boko, and Jocelyn were the drivers of all the blood work. And Robert Padera is my close friend and collaborator. We teach together in HST. He runs the autopsy service at the Brigham and Women's Hospital. So we have, we are in regular contact on this stuff. Without this involvement, I wouldn't have done this work. And there are many other key contributors at the Reagan Institute, and I can't name them all. Everybody who from the graduate students who went in during the COVID outbreak, when we had lockdown, and went in just to serve as technicians to process blood every day and to get the flow done every day. I mean, they were heroes. I tried to put them all on the paper. A lot of them were grad students, some were technicians, some were postdocs. They all volunteered to do this. So this wouldn't have happened without them. And of course, the patients we owe a huge debt to.
Thank you. Thank you very much. Great data and everything just come up. So I think we should open it as a panel discussion. I'll bring out some questions and I'll invite Dr. Mehra also to join in if at all he's around. I see him around. So
I do have one question for Narinder, which I wanted to
okay,
ask
please
before.
go ahead. Go ahead.
That question was, I wanted his opinion on this. So, there are only two diseases in which GWAS that I know of, where you would expect GWAS to give a signal from MHC, and it doesn't. I think the explanation in IBD that you don't get a GWAS signal, there are so many different microbes in the gut that every MHC can work. So it doesn't matter, you don't get MHC signal. All other inflammatory diseases, you get MHC signal. But in COVID, in the big GWAS studies so far, there hasn't been an MHC signal. And I'd like to get your opinion on that.
I agree with you on that. And that is why I said that there is a... Sunil, can you allow me to have my camera on? Because you have
Please.
blocked me. I can only speak, but it's good to see faces.
I think all panelists can open camera, but I'll ask.
Your video is just click on it.
It says you cannot start your video because the host has stopped it.
Okay, I'll ask Anupam to kindly start camera for Dr. Mehra, please.
You are very right, Shiv, that there is not a single study done so far on COVID-19 on those. That is why a genetics consortium has been made, and they've been also asking us in India to be a part of that. There are some issues on that actually. So one of the things that they want to do in that is exactly the kind of thing that you're talking about. It's not just an association one is looking at. It's not just that who are the people who will go on to develop a milder form or a more severe form, because the issue is patients who get hospitalized, who are the ones who will come back home and who will go up. So I think that's the issue, and the feeling is that you could look into. So it's not just an allele here, but because you could see COVID-19 also as an inflammatory syndrome, much like any autoimmune disease there. So people would look at the haplotype. So the NGS-based typing would also help to define some extended haplotypes of an MSC1 or an MSC2. At the moment, I agree there's no.
So I believe the haplotypes will show things because the locus controls so many other things beyond T cell activation. I think overall the signal for T cells is weak so far.
But I may tell you here, when we are talking of an autoimmunity favoring haplotype, which in the West and the population that you deal with is only one which we call ancestral haplotype 8.1. It is a HLA-A1, B8, DR3, and then the complotype it is SCO1, and goes on, and it is a characteristic haplotype of the Caucasian. This haplotype does not exist in India. We have multiple actually DR3 positive haplotypes which confer susceptibility to autoimmune diseases. Gaurav, who actually was in my lab, he's now with Sunil, he could actually give you a list of those 8 or 10 haplotypes that we know of. It is not just the B8 DR3, but we have others as well. So the diversity of even the autoimmunity favoring or the inflammatory supporting haplotypes is much more in the Indian population.
Happy to answer other questions, Sunil.
Okay. So there are some questions which you said you will explain it. You have already done that. What was antibody titer, IgG, IgM, and you said I will try to explain. You have done it. Any comment on NK cells during COVID infection?
So the evidence, unfortunately, is that NK cells, though they go up with age, they decline dramatically in COVID patients, like all lymphoid populations. So in the lung, again, you won't find them. They're not, if you hope for protection from them, it's not happening.
Okay.
Probably in mild
So
disease important.
do you see any role of interferon gamma in COVID-19 infection? We always talk about this cytokine for viruses and intracellular pathogens.
So, I mean, the role I am seeing so far is not a good role, of course. It should be protecting you from virus. I think we are seeing increase in Th1 in the lymph node. In relative proportion in the lung, there's some Th1 late in disease, a very small number. This lymphopenia is severe. So maybe Th1s are making the disease worse at that point. Uh, at the time when you're mild disease and you're protected, there's hardly any evidence for any T cell activation to begin with. So there might be some interferon gamma being made by innate cells at that time, but that's not what you need, right? At that point, maybe interferon gamma might activate your NK, and that could be so, but evidence is very limited.
All right. The another one is that what are your comments on antigen-specific T cells in SARS-CoV-2 in unexposed individuals, as reported by
Shane
Shane
Crotty.
Crotty?
Yeah.
Yeah.
So I actually think that because we all get common cold coronaviruses, and unlike RBD, which is so specific and unique for humoral immunity, there are so many T cell peptides which are cross-reactive between the other common cold coronaviruses and the severe viruses too. So most people have some T cell immunity. I mean, and the T cell immunity lasts for a very long time. So if you got some common cold coronavirus, or you even got SARS and survived it, you probably will make T cells that will be there for a long time. Do they protect you from infection? Not clear. No evidence. Maybe they'll attenuate disease. I'll hope so. The studies have not yet told us what Shane's latest paper suggests is that older people have less of the responses that you want to see that you see in younger people. So older people, anyway, we have depletion of T cells and all with age, and so you have less of a naive population. And so yes, in an older individual, you make less of a response during disease. That much is clear. But in mild disease, so if you get moderate disease, you will get a T cell response. Does that mean it's protective? That's no evidence. I'm telling you, there's so many types of T cells, so many types of responses. The way the immune system works is that if a pathogen persists, you will get every response. Which one of them is protective? Which one of them is helping you? Or which one is making the disease worse? That's what you want to tease out. So at this point, just like for the CD4 CTL data, because from yesterday to engaged, we're very interested in CD4 CTL. CD4 CTL is extremely rare in the lung. So I don't think it's, first of all, protective or causing disease, as far as we know. But yes, we see them in the blood. There is no question. But does that mean something? I don't know.
So somebody wants to ask you a question regarding reduced virulence, which is being speculated of the virus, that virus is losing virulence over time. Is it true or no?
I think there's no evidence for that. So as at this time, that means you would have to find a viral mutant with, let's say, a mutation in NSP3 or NSP1, NSP10. That hasn't been shown. What has been shown is some differences in the spike so far. So as Joshi pointed out yesterday, if there's immune pressure, with time, maybe the virus will mutate the spike, the RBD. But keep in mind that if the virus mutates RBD, and it can't get into your cells, if it goes to that level, that's a problem. But because we can make antibodies, the monoclonal antibodies that have been made are against a couple of domains, one next to the RBD and the RBD itself. So you would think that some of the vaccination strategies will allow us, by steric hindrance, to actually bind to sites near the RBD neutralization site as well and cause a blockade of infection. So I'm actually hopeful vaccines will work pretty well when they come out. If the virus evolves and we have a problem a few years from now, let's deal with it then. Okay, we'll have to think that through. But this is not a highly mutagenic virus. It is not going through recombination events like flu. Its error rate is low in terms of when it replicates. So in many ways, it's not designed to evade us that badly.
So what's your take on that in any particular community, there is a peak and then slowly the infection rate goes down. So what is that coming up? How do you explain that?
So actually, yeah, there are many explanations for that, which I'll suggest a couple. One is, of course, so I thought the Dharavi data showed that more people got infected, and then after that, so Jayaprakash Mulil, who's my classmate from medical school, had suggested to me that there was a high antibody rate coming up in some patients in Dharavi. My assumption is this: you do make some antibodies, okay? Some people make antibodies. They last for maybe, if they last for 3 months, it's good, right? So in the people who made those antibodies, they're all coming from the extrafollicular area. That's what I think. You're making neutralizing antibodies which are class switched, and they're coming from an extrafollicular site. They may not last very long. Maybe hopefully the last 6 months or something like that, at least for 3 months in many people. That will attenuate spread, reinfection, and so on. So people are getting subliminally infected. Maybe a little bit of that is going on. Maybe as the number of targets reduce, the people who expose themselves, maybe something as simple as the people who expose themselves most got infected early, and then the people who don't expose themselves too much don't have people to infect them after some time. And there could be some reason like that, but I don't have an explanation.
Okay. So one question related to CD8 cells level and prognosis says that there is a recent paper from China, which says that they are being inversely proportional, irrespective of the antibody response. So in light of this, should we not look for the vaccine that is based on T cell epitopes rather than looking for antibody response?
So one of my close colleagues, I think, he does very nice work. His name is Gaurav Gaiha, who was once upon a time my student. He's now a faculty member at the Reagan. He published a very nice paper last year in Science. I don't know if you saw it, looking at networked peptides from HIV to try to find those peptides which you cannot afford to mutate, and based on which he's trying to design vaccine. He's trying to do the same thing for CD8 cells in SARS-CoV-2. My only concern is that there is no evidence to date that a CD8 type vaccine protects you from infection. It should protect you after you're infected by helping you maybe to clear virally infected cells. So yes, it's not bad to have a CD8 component, but if you want to protect people from infection and not, remember, if you get infected in the nose, and even if you don't get severe disease, you're going to spread the disease, okay, to other people who may not have got vaccinated or whatever. So I think, yes, maybe having a CD8 response is not a bad thing. Again, there's no evidence it protects. Certainly no evidence that it is driving severe disease. And there's no evidence that any CD8 vaccine currently is key to protection from any known viral disease.
Okay. I think we should restrict, because there are so many questions. I'll pass it on to you via email. So we cannot ask all the questions. We have next speaker.
Hey, can I just ask one question from
Yes,
Shiv?
please.
He referred to the reinfection, and he feels that the reinfection would be possible. Of course, it would be. But so far, 9 months down the line, we haven't seen many cases of reinfection. And the one that we saw from Hong Kong was a guy who really traveled and by accidentally, because once you arrive at the airport, you need to have the test done, and the test showed that he had second infection. But would you feel, even if there's a reinfection, would you feel that the reinfection, the guy would be having a more severe form or a milder form or asymptomatic form?
Yeah, I mean, we should all hope that people don't get sick and have severe infection, right? So Michael Minna at the Brigham has already found a few cases of reinfection. So it is happening, obviously not at a very high rate. Reinfection, people are protecting themselves more. The proof of reinfection always comes from sequencing the virus. I mean, you cannot prove reinfection unless you resequence the virus. So far, I don't see, because the numbers are so few, I don't see evidence that you get severe infection the second time. But keep in mind that 80% or even higher percentages in India don't get severe disease, right? So people tend to discover reinfection. It's theoretically possible. We don't know the true incidence of it. I hope it's not high.
No, I'm not talking of India. I'm talking globally. do we see even in Europe, which is now seeing a second wave kind,
Yeah.
see reinfection?
But, Narendra, keep in mind, this is only a few months. The first wave was a few months ago.
Yeah.
So the worry about reinfection is not now. The worry is if there's some joker who believes in herd immunity from natural infection, and 1 year later, will he actually be protected? Of 2 years later, right? That is the worry I have. It's not about somebody in 3 months and 4 months. You're probably protected for some time, okay? Let's assume that. But it's the longer term thing. So if you don't get vaccinated, my worry is that you will hope you're protected. There are people in our country with stupid people who have been holding corona parties. This was happening earlier on in the hope that they'll get infected and then they'll be protected, right? That was the kind of thing that was going on at one time. I think those things you have to discourage, because we are not confident that, a year out, year and a half out, will you still be protected? No evidence for that. And all the indications from looking at the immune system and the response is that that might not happen. There is reason to believe that.
All right. Can we stop now here and wait for another? Shiv, are you going to be around for another?
I have to teach a class at 10. That's the reason. Rama, I'm going to miss a little bit of your talk.
Okay.
I'll hear a bit of it, but I'm happy to listen to him.
Anyway, great to see you, Shiv.
Thank you very much. Thank
Great
you.
talk.
Rama, I need to introduce you. We have the next speaker, Dr. Rama Amara Rao, who is from Emory University, Georgia, Atlanta. Rama is Charles Howard Candler Professor in the Department of Microbiology and Immunology at Emory University Vaccine Center. He works at National Primate Research Center of Emory University. He received his PhD from the Indian Institute of Science, Bangalore, India, and did his postdoctoral fellowship at the Emory University. And then his research is focused on development of vaccines for HIV, tuberculosis, SARS-CoV-2, all these viruses. So then Amara's lab has pioneered the heterogeneous prime boost protocols using DNA modified vaccines, Ankara, and novel protein immunogens for vaccine delivery. So I'll leave it to you, Amara, for Rama, to carry on with your talk, and we listen to you on development of this vaccine. Amara, stage is yours.
Thanks, Sunil, for that wonderful introduction. And also thank you and organizers for giving me this opportunity. I see two Shiv pillais here. One is smarter than the other one. I see the Joshi's name is now shows as Shiv Pillai. So the other Shiv has more hair than the actual Shiv. Anyway, I just wanted to point that out. Anyway, good evening to all of you. I'm sure it's getting to your dinner time, so I try to finish it as fast as possible. So the way I structured my talk today, so we had done a lot of vaccine work on COVID-19. A lot of it is being put together for a manuscript, which is about to go out this weekend. So there are some that we have already put out on the internet. So I'm going to be talking about those data. But what I thought was that I could take this opportunity to give a little more general view of vaccine development. Primarily what I learned over the last 20 years. This is more of a practical knowledge that I could share with the community in India. That way, that would be useful for the general vaccine development. So these are the top 10 global causes of death in 2016. You can see the heart disease up there, and the stroke, and the pulmonary disease, lower respiratory infections, cancers, diabetes, diarrheal diseases, and TB. So the point is that then infectious diseases do contribute for the global death in a significant way. And then these are multiple infectious diseases that need vaccines. Respiratory infections is up there now with the COVID. This number is going to increase dramatically more. But the infections where we have vaccines, the point I'm trying to make is that those infections are very, very small compared to the other infections for which we don't have vaccines. And there are, just to remind everybody, that there are a lot of cancers that are caused by infectious diseases. If we make vaccines against these, you could also impact the cancers. So just coming to the basics. So when we talk about vaccination, what are we talking about? So the vaccination is the deliberate induction of protective immunity to a pathogen by administration of non-pathogenic forms of the pathogen or its antigens to induce a memory immune response. So the memory is what is key here, what we are trying to generate by vaccination. So this is the picture of Ed Jenner way back vaccinating a kid for TB. You have seen this, multiple times. And this is the experiment that we won't be able to do it now. So how do these vaccines work? They work by mobilizing memory immune responses. They protect the individual by limiting the infection so that it does not cause disease. They protect the population by limiting transmission. So the vaccines work by antibodies. So in the antibodies, you have neutralizing antibody and non-neutralizing antibodies. The neutralizing antibodies, they block the virus entry to the target cells. They inhibit the binding of the virus to the receptor on the target cells and neutralize the virus. And then they also have multiple non-neutralizing antibody functions like ADCC, ADP, now ADCVI. So all these functions also play in a role in antibody-mediated protection. The vaccines also work by CD8 T cells through their cytotoxic function. It requires MHC class 1 presentation of foreign peptides. They also secrete cytokines, chemokines, some other factors that can actually induce suppressive functions. So all these are important for mediating vaccine-mediated protection. And you also need CD4 help, which is central to both of these arms of the adaptive immune response. So they help CD8 T cells to generate functional effector cells that induce a central memory response. And then they also help B cells in the germinal center formation. That's where the affinity maturation of the antibody happens, and that is critical for inducing neutralizing antibody response. And the CD4 help comes in different forms. You have Th1, Th2, TFH, Th17, Tregs, and T follicular regulatory cells. And then there are more now, like Th9, and the list is growing. So these different helper cells, I'm not going to go into these because you might have already heard, they help against different infections. So coming to immunological memory, how the vaccines work. So here is the infection that takes off. This is an example of an acute infection. There is a fast growth of the bug, and then the body mounts a primary immune response that comes a little late. It generally takes about 1 to 2 weeks. And then this is this is comes at a low level. But when you have a memory response where you start with some pre-existing frequencies of memory cells, and that they rapidly expand, and then they clear the virus, and then they maintain a memory response at a higher level. So basically, the memory cells, they start with at a higher frequency, and then they also respond more rapidly. For example, they can divide within 6 hours as opposed to 16 to 24 hours of a naive cell. So to initiate a vaccine response, you need antigen to provide specificity and memory. For example, cytotoxic cells require synthesis of antigen in the cells. T helper cells require antigen, external antigen, and the antibody requires external antigen as well as antigen synthesized in cells. And the innate immunity is to provide danger signals. So both are important. So there are multiple steps in the what that happen during the priming and immune response. So you have maturation of antigen-presenting cells. They take the antigen, they drain to the draining lymph node. That's where they either prime the T cell response or a B cell response. And there's some T cells that leave the lymph node and go to the site of priming in search of the infection or the antigen. And then you also have T follicular helper cells. They go to the B cell zone and then help the B cells. So these are general steps that happen during vaccination or any antigen priming. So the antigen can be delivered as multiple forms. It can be recombinant viral or bacterial vectors. These can be live attenuated vaccines or killed whole vaccines. The purified protein or polysaccharide-based vaccines, conjugated polysaccharide-based vaccines, DNA vaccine, RNA vaccine, and you can also use a combination of any of these 2 vaccines or 3 vaccines. So these are called heterologous prime boost vaccines. So you must have heard about almost all of these during the COVID pandemic, that now there are a number of vaccines that are being developed, and they're using one of these different forms of the vaccines. So why do we need this many vaccines, and how do they differ? So they differ in terms of the immune responses they induced, whether the antibody response or CD8 T cell response or CD4 T cell response. The live attenuated vaccines generally give you everything. And then they also give you a Th1 biased CD4 helper response. And the killed whole vaccines, they give you pretty good antibody response, but very poor CD8 T cell response, generally a Th2 biased help. And the proteins and polysaccharides based vaccines, they give you antibody, again, poor CD8 and Th2s. And basically, when you go to the DNA, that you get, again, all 3 components, but at a very low levels. So there is a low level of priming. Now this can be increased by electroporation and things like that. And if you use a heterologous prime boost vaccines, for example, let's say DNA prime and a recombinant viral vector boost, you can actually increase very high levels of all 3 components of the immune system. So, and then they also have some disadvantages, like live attenuated vaccines. If the attenuation is not proper, or there is also an issue, that could be an issue of reversion to virulence. There could be contaminating live agents. And the killed whole vaccine, if there is an incomplete killing, that is a problem. The purity. Proteins and polysaccharides, they need generally adjuvants. In the conjugate vaccines, T-independent to T-dependent T cell responses, generating responses in children under 2 is generally challenging. Recombinant viral vectors, there is an issue of pre-existing immunity. The DNA, there is a built-in adjuvant CpG, and the heterologous prime boost. These are very powerful, but they require two agents. So when you have to make two vaccines, it can always be challenging. So these are some live viral vectors that have been used. This is the adenovirus, this is MVA, this is polio. So the different vectors have been used. Again, when you have a choice of different vectors, how do you choose? So the first thing is that you want to make sure that the vector has the excellent safety record. And then in terms of the immunogenicity, it should induce high magnitude, quality, and long-lived immune response. And it should be able to carry large recombinant inserts. And it should be easy to manipulate so that you can make different recombinants pretty rapidly, and less pre-existing immunity. And it should have high titer growth in vitro so that manufacturing is easy. And it should have high stability during manufacture. So that these are the different vectors, and then for their examples and various qualities of these, and I'm not going to these details. And then recently, I want to point out that cytomegalovirus 68.1 is being developed as a vector for HIV, TB, and other indications. This particular vector is unique in the sense that it induces MHC-E and MHC class 2 restricted CD8 T cells, as well as supertopes. These are the epitopes that the same epitope can be seen across multiple individuals. It can induce long-lived T cells in the mucosal tissues. And then there is some nice protection data for SIV and TB in monkeys. So I want to point out that the 4 important goals for vaccines: they should induce high magnitude immune response, right quality immune response, and the location of the immune response that is induced is very important, and the persistence of the immune response is very important. So these are regulated by the combinations of the vaccines that you use. If you use a heterologous prime boost, which is best for inducing everything, I will touch upon this a little bit later. In general, for T cells, inducing T cells, a DNA or live vector prime and the live vector boosting is the best. For antibody responses, DNA or live vector prime followed by the protein boosting is the best. So one would have to choose based on what is the kind of immune response they're trying to target. So we worked for the past 20 years in a heterologous prime boost approach using DNA prime, and the pox virus is an MVA carrying the insert of various infections. And we showed that you can actually induce very balanced high levels of T cells as well as antibodies. Here is an example. So if you immunize monkeys with MVA vaccine, after the priming, you get reasonably good CD8 T cell response, but the subsequent boost doesn't really boost this response further. Whereas if you prime with the DNA and then come in with a single MVA boost, then you get 10 times higher CD8 T cell responses. So this is just an example of how the homologous prime boosts, the heterologous prime boosts are superior to the homologous prime boost. And recently we have been working with heterologous viral vectors, for example, VSV prime, Vaccinia boost followed by Ad5 boost. So you can use this kind of approach to induce very high frequencies of CD8 T cells. Here, in some animals, you can go up to 60% of the CD8 T cells are specific to that SIV gag that what we used. And then we recently showed in a paper published that when you combine this heterologous viral vector approach with a protein-inducing vaccine for HIV, we can get durable protection from infection even 1 year after vaccination. So we are now trying to understand. So one of the reasons that this works very well is that we induce these CD8 T cells that are in very different compartments of the body, including liver, lung, ileum, jejunum, colon, rectum, everywhere, even in the female genital tract. Very high levels of CD8 T cells. So these are tissue resident memory T cells. So we are trying to understand the ways to induce these cells. These cells sit in the local tissues, and then they can fight the infection at early stages so that those infected cells can be rapidly cleared and provide protection. And here I'm just showing an example that how you can actually boost the antibody response by adding a protein boost to a DNA MVA vaccine. And here there are two groups that one group got the DNA MVA vaccine, which is in teal here. When you come in with a protein boost after 2 MVA boosts, that you can boost these antibody titers by about 40-fold. So it is a powerful way of strongly boosting the antibody response. And Shane Crotty's lab has shown that you can actually use these pumps where you can release the antigen slowly over time. That can dramatically improve the ability of the vaccine to induce neutralizing antibody response against HIV envelope. And I'm sure that the more work is being done to make this a practical approach. As a proof of concept, it really makes an important point. So then comes to the point of adjuvants. These can be used to enhance the magnitude of the immune response. There are multiple adjuvants have been used. I have named only a few here. Co-stimulatory molecules have been used adjuvants like CD40 ligand, 4-1BB ligand, OX40 ligand. The cytokines have been used, and the toll-like receptors have been used as adjuvants. Agonists. So they basically activate your innate cells to lead to the better priming by enhancing the co-stimulatory molecule expression as well as the production of cytokines or chemokines. And some adjuvants work at different levels. For example, GM-CSF can recruit monocytes, promote their differentiation and maturation, and then also inducing T follicular helper response. So each adjuvant works at different levels. And then there are multiple toll-like receptors that can act as adjuvants that they deliver different kinds of helper response. So coming to the quality, not all vaccines induce the same kind of immune response. For example, that looking at production of interferon gamma, IL-2, and TNF, one can define 7 subsets of T cells that are co-producing cytokines. You have cytokines that you have cells that express all 3 cytokines versus any 2 cytokines or any 1 cytokine. So these we call as triple producers. And here is an example that T cells induced by different viruses in humans. They're different. For example, vaccinia versus influenza-specific T cells or CMV-specific cells. So the vaccinia virus induces the highest levels of triple producers, whereas CMV is less. Whereas the CMV specific cells produce a lot of interferon gamma and TNF. So these can change depending on which virus is inducing these immune responses. And this makes a point that using the dose of the vaccine and the route of the vaccine is important. If you using high dose is not always necessarily true and necessarily good. For example, here the high dose intramuscular immunization induces less triple producers than the low dose intramuscular immunization. And then there is the sub-Q immunization induces even higher triple producers compared to the intramuscular immunization. And then this reflects in the ability of the vaccine to protect. This is an example using the Leishmania major challenge. The vaccines that induced higher levels of triple producers, they actually show better control than the vaccines that induce lower levels of triple producers. Although the vaccine was used at higher dose, it was less beneficial. And in fact, this has been also seen for some of the experiment papers where they used RBD protein for COVID-19, where when they used with a strong adjuvant with a higher dose of the protein, in some cases they completely lost neutralization titer. So the dose combined with the adjuvant needs to be carefully defined for any vaccine. And the different vaccine vectors prime different kinds of memory cells. Broadly, these are defined as central memory that survey the lymph nodes, and the effector memory, they survey the non-lymphoid tissues. And here is an example that taking the HIV gag, either put it into AD5 vector or MVA vector, and then if you watch the kind of memory response that is generated, the MVA generally gives you higher central memory response, where the AD5 gives you less of a central memory and more of an effector memory response. So depending on which vector is driving this, there would be a difference in what kind of response we're going to get. So the priming and boosting required for formation of memory cells and affinity maturation of the antibody is critical. Live viral vectors typically require one immunization. Inactivated viruses or protein-based vaccines require repeated boosting. And then it is important to space out this interval so that there is an affinity maturation of the antibody that happens over time. As the antigen becomes limiting, you select for high affinity B cells, and then you boost the response once this has occurred, so that you induce high affinity antibody response. That is important for generating a strong neutralizing antibody response. So it is important not to boost response very frequently, like every 2 weeks or every 4 weeks. Typically, it's good to wait at least 2 months. And in terms of the location of the immune response, this is regulated by various molecules that are involved in regulating the trafficking. And for example, in alpha-4 beta-7 helps in trafficking to gut, and CCR9 to small intestine, CCR10 and CL8 homing to skin, and CCR5 is homing to tissues such as liver, lung, and genital mucosa. And this imprinting on these T cells is done by the DCs that are present in that tissue. Here I have shown some examples for like vitamin A can imprint homing to the small intestine because that this is regulated by retinoic acid, which can drive the expression of CCR9 on the T cells. And when the DCs are priming these T cell response in the draining lymph node, if these DCs don't have ADH and RALDH enzymes, they can't convert the vitamin A to retinoic acid. So only the DCs that are present in the intestinal tissues, and they express high levels of these enzymes. That's why they're able to prime T cells that home back to the intestine. So similarly, so if you actually do a systemic vaccination and in the presence of retinoic acid, you can induce T cells that home to gut. So generally, the DCs that are present in the systemic compartments, that they don't express the RALDH enzyme. That's why they can't imprint T cells that home to the gut. If you provide that exogenously, you can do it. And then we also showed that some time back that viruses like AD5 and also MVA that can induce these vitamin A metabolizing enzymes in dendritic cells in the peripheral tissue so that they can induce these gut homing CD8 T cells. And this is just data that was published in this area. I'm not going to those. Similarly, vitamin D3, the UVB, can imprint T cell homing to the dermis by induction of CCR10. Again, you need these enzymes to convert to make this 1,2-dihydroxy-3. So this homing of this immune response can be regulated by modulating these factors. So now I'll come to our COVID-19 vaccine development. And we aim to generate both antibodies as well as T cells because we think they both are critical for protection. As I said before, the neutralizing antibodies block infection, and the CD8 T cells can clear those infected cells, and the CD4 help is critical for both. So when we think about COVID-19 vaccine, so these are the things that we again think that long-term safety should induce both antibody and T cell responses, induce immunity in the lung if possible, should be able to provide cross protection. So our long-term goal is to design a vaccine that can potentially protect against multiple human coronaviruses. And then the immunity should be long-lived. So we are working on three different approaches: the MVA-based vaccines to induce strong neutralizing antibodies, CD4 and CD8 T cell responses, and the protein-based vaccines. These target the inducing neutralizing antibody responses. And then the T cell plus antibody-based vaccines. Here we are using the heterologous prime boost vaccine approach. So this is the lab and the people that contributed to the work. This is the MPA vaccines are generated by Shailaja Gangadhara and Nanda Kishore Rautu and Narayana did the animal experiments, and Satish is doing a lot of innate analysis in the NHP model. And these are done in close collaboration with Mehul Sutar's lab at Emory. And Abdul Rahman is doing beautiful in immunohistochemistry work. Unfortunately, I can't show you all of those data, but I'll show you some. So this is the initial work with our MVA vaccine we published in bioRxiv, but we have done tons of work now on this, which is unpublished. It's a beautiful work, and which hopefully will come out pretty soon. So the MVA was originally developed as a vaccine for smallpox for use in immunocompromised people. It was derived from vaccinia virus by simply passaging many times in chicken embryo fibroblast. During that process, it underwent 6 internal deletions and became replication defective in primates and lost most of its immune evasion genes. And it has an excellent safety record in humans, including in HIV-infected people. So there are so many advantages for the MVA vaccines. So they're safe, well tolerated, induce strong antibody responses after single vaccination. The immune responses induced are durable, and we have shown in humans that they undergo very little contraction over a 6-month time frame. And in the monkeys, we have shown that we can follow up to 3 years that the antibody responses are pretty stable. It can be delivered through multiple routes, including intranasal vaccinations. It can accommodate large inserts, and then the recombinants are highly stable and be used for the vaccine manufacturing. And then use both CD4 and CD8 T cell responses. And importantly, the MVA-based vaccines have been shown to protect against SARS-CoV-1, MERS-CoV, Zika virus, and Ebola virus. So based on all this background, we started making MVA recombinants using spike protein as the target to induce neutralizing antibody responses. So here we made 2 versions. And one version, we express this entire spike protein that includes both S1 as well as S2 and the transmembrane and the cytoplasmic tail. And we made another recombinant that expresses only the S1. In the full-length spike, we introduced these pre-fusion stabilizing mutations so that the protein tend to stay in the pre-fusion status. So these are the 2 constructs. These are MVAS that express the full-length spike with the pre-fusion stabilizing mutations, and then the MVA-S1 that expresses only the S1 region. So when we immunized these mice, then we saw some important differences. First, when you look at the binding to S1 or the total S, the MVA-S1 protein, which is actually secreted out of the cells, induced higher S1 binding antibody compared to the MVA-S. But when you look at the total S binding, the MVA-S induced higher binding than the S1. This is expected because you don't have antibody response to S2 in the MVA-S1 group. However, when we look at the binding to the RBD, where this is the antibody that contributes to neutralization, while both vaccines induced RBD binding antibody, the MVA-S induced a little bit higher than the MVA-S1. Actually, surprisingly, that when we looked at the neutralization against the live SARS-CoV virus, only the MVA-S vaccine induced antibodies neutralized the virus, not the MVA-S1 vaccine induced antibodies. And then we saw, although they do have pretty significant binding titer to MVA, RBD, as well as S1. And we are investigating this, why these antibodies don't neutralize. Obviously, the specificity of these is going to be different. And we see a direct correlation between binding antibody to RBD and neutralization, and actually an inverse correlation between binding to the S1 protein and neutralization. Both vaccines induced comparable T cell responses. If at all, the MVA-S1 vaccine induced a little bit better, although they are not statistically significantly different. We saw both CD4 and CD8 T cells. And then we also saw these inducible bronchus-associated lymphoid tissues in the lungs. Those were seen more in the MVA-S vaccine compared to the MVA-S1 vaccine. I don't have the data here. We also have seen the S-specific T cell response as well as S-specific binding antibody in the lungs of these mice. And this shows that the MVA vaccine-induced immune responses can go to lung. These are the responses in the lung. I do have it. I thought I don't have it. So you can see that there is nice binding, good CD8 and CD4 T cell responses in the bowel. And the ongoing studies, we immunized rhesus macaques, and we showed that we can get the similar antibody and T cell responses with MVA-S vaccine. And then we also challenged these monkeys with the SARS-CoV-2 and then showed that we get great protection. We have very nice immunology done in the monkeys in the lung. So all those data hopefully would come out pretty soon. And in terms of the protein-based vaccines, we made monomeric and trimeric forms of the protein. Then we immunized mice, and then we show that you can induce pretty strong neutralizing antibody titers using these protein-based vaccines that are quite stable, at least in the mouse. And by using different adjuvants, here is an example of adjuvant 1 and adjuvant 2, you can induce pretty strong neutralizing antibody that is, in several thousands. And right now we are testing these proteins and adjuvants in the monkeys, and these monkeys also will be challenged pretty soon to look at the protection. And then we're making CHO cell lines expressing these proteins with a path for clinic. So that's where we are with the COVID vaccine development. We are right now actually manufacturing the MVA for going into clinic. So hopefully we'll be able to go through the clinical development pretty soon. So I'll stop here and take any questions.
Thank you very much, Rama, for overview of how to, what kind of vaccines we should, how they are developed and how they have efficacy. Before I open the question answer box for you, I need just one clarification. I read a very nice paper where they have used, what is your route of immunization actually? How are you doing?
So we are using the intramuscular vaccination. Okay. And then in the mouse, now we are testing a combination of intramuscular and intranasal vaccination.
Yeah, I think you know about this paper, which shows that the same vaccine, if it is given intramuscular, is not effective. While given nasal immunization, it brings in so much of efficacy increase. So the route of immunization might be really important for this,
Yeah, in our experience, when we were doing this for TB, I think if you do simultaneously both IM and intranasal, that you can actually induce very high frequencies of T cells in the lung.
Yeah.
We could go up to 30–40% of the TB specific CD8s in the lung.
Okay, so there are a few questions for you regarding this vaccination. This is one question by a student who wants to ask you, in case of T-cell based vaccines, how much does the individual T-cell epitopes quality affect the vaccine efficacy?
I think it plays a very important role, and it depends on what kind of helper response that we would need against that particular infection. Regardless, I think it is important to have a T follicular helper response. And we have shown for HIV that vaccines that induce very high levels of Th1 response, we published this earlier in STM, that vaccines that induce very high levels of Th1 response, they express CCR5 and migrate to the mucosal tissues. And then they could actually sit there as tissue resident memory CD4s. And then they can influence the protection in a negative way. So, and then subsequently we have shown that this part is not published, that if you change the route of immunization, then you can change the balance between Th1 and TFH response. Thereby you can actually modulate the protection. So it is the vaccine delivery route can impact the T helper response that could impact the protection.
Yeah. So this, I think he's trying to develop a multi-epitopic construct for as a vaccine candidate. So he asks that can epitopes added together synergistically to boost the vaccine response?
Yeah, so yeah, you can stitch together multiple epitopes, but then you have to pay attention to the protein folding, and not these things are not straightforward. But one thing I would say, based on our experience, is I would use intramuscular immunization over intradermal immunization. Intradermal immunization doesn't give you a strong T cell response. So I would stay with, if you're going with a systemic immunization, I would go with intramuscular.
So there is another question which says that for vaccine efficacy, can we use polyfunctional triple positive T cells as indicators, and at what time period can we expect triple positive cells to appear in the in vitro cultures?
Yeah, generally these are you have two phases mainly after vaccination. You have an effector phase and then you have a memory phase. So this polyfunctionality should be defined more in the memory phase. In the effector phase, the lot of these T cells don't produce IL-2 because their job is to basically kill at that point. So I would wait a few weeks after immunization and then measure the quality of the T cell response. And then in general, the polyfunctional cells are superior than the less cells that are making only interferon gamma for protection against multiple infectious diseases.
So Rama, there is a question regarding monoclonal antibodies which have been given to President Trump. So do you have any idea what kind of those antibodies were given?
Yeah, so I haven't followed carefully, but there are two antibodies that target, these are neutralizing antibodies. And I was, the moment I heard that they were going to give him that, my first response was he was going to live. So I have a lot of faith.
You did not want him to be.
So I told my wife, "Oh yes, he's going to live." I think monoclonal antibody therapy is one of the powerful therapies for COVID-19 disease or infection. I think that should be really pushed forward and should be fast tracked.
Okay, there is one question directly about your modified Vaccinia Ankara. So how MVA is better than ADV5 vector inducing immune
And actually, we did a head-to-head comparison. AD5 is actually more immunogenic than MVA in terms of inducing the T cell responses. We haven't carefully compared the antibody. Antibody, they may not be that different. But the AD5 issue is with the pre-existing immunity. So which we have less of a problem with MVA unless you're going into people that are older than 60 or so. And even with AD5, they're thinking that if you do an intranasal vaccination, you may be okay. And also AD5, I think in general works well for intranasal vaccination than MVA. I think the main issue is the pre-existing immunity with AD5.
So somebody is asking, why did you choose viral spike protein? Why not any other viral molecule?
This is something we started way back in March. That time there was little information available. And we modeled this based on the data that was available from SARS-CoV-1 infection. Because the spike of the CoV-1 and CoV-2 are pretty identical for most part, except for the RBD region. And now we have designed vaccines that are chimeras of various proteins of SARS-CoV-2 to induce T cells across different proteins. So those are under development, and then so we do have other MVA recombinants that target other proteins of the virus.
So, what do you think? What kind of adjuvant would you like to use to have the, uh,
For the protein?
skew of TH1 or TH2 type of,
Yeah, so one thing that we found so far is that alum itself is quite good. So in general, the spike protein is super immunogenic. So compared to my experience with HIV envelope, the spike protein induces very strong immune response. And in the mouse, the immune responses, they persist quite well just with alum. But mouse is not the right model to study the persistence in general. So we now have the NHP experiment, and there we'll know more. Immune responses contracts a lot more in humans compared to mice, and the NHPs kind of give you some better idea of the contraction. And there are a lot of other adjuvants that are being tested, like GSK is looking at ASO3. And we are looking at 3M adjuvants, which have shown induced very high neutralizing antibody responses. So I think all these adjuvants like MF59 or Adavax or ASO3, these are all TH1 inducing adjuvants. In general, that adjuvants that induce a stronger CXCR3 positive TFH, they seem to give you better persistence of the antibody. I think the key thing here is to see how these actually fare when individuals are exposed to the virus. So we don't have any data yet to answer that question. So one need to be very careful with these protein vaccines or adjuvants.
Yeah, although it has been shown that it's not as rapidly mutating virus as the flu viruses, but there is a question regarding this only. So if you select a protein, and what is your take on, like, how long it will be sustainable, the vaccine which you have produced? Suppose the virus comes back with a mutated kind of.
You definitely we may definitely need to go beyond alum in that respect to induce persisting antibody responses in people. But we need more data to make sure that these adjuvants don't backfire. Once there is there's no over inflammation in the lung when these individuals are exposed to the virus. But if in general, if there is a strong neutralizing antibody response, then we should be okay because there is less replication of the virus. So that should not induce strong inflammation.
So among monomeric and trimeric form of protein, have you found difference in immunogenicity?
Yes, so those are ongoing right now, but we initially tried with the 10 microgram dose and we did not see a difference. Both were equally good. When we went down to 1 microgram, we start to see a big difference. So it looks like early results that you can actually induce much higher neutralizing antibody with lower doses of the protein if you have a trimer.
Okay, there is one question on, can the antibodies generated against a particular epitope rise in different species of animals can have different binding affinity for the same epitope?
So using the same immunogen in different species with the...
Yes, yeah, it can be, yeah.
I think I've seen that even in mice, with the different strains of the mice, the level of neutralizing antibodies induced by vaccine are different. And my feeling is that you get better neutralizing antibodies in probably in rabbits and guinea pigs than in mice. So each same immunogen, its ability to induce neutralizing antibody seems to vary quite a bit between different species. Um, so yeah, so that that needs to be carefully looked at. But I don't think anybody has characterized the affinities of these antibodies in different species, and I'm pretty sure there will be. And also, one thing to keep in mind is the antibody subclass is studying that is going to be tricky. For example, in rabbits, that you don't have this class switching. You don't have these multiple Ig subclasses, and they can influence the effector functions of the antibody. So, each model is good for different things.
So yeah, whatever response you get in an animal model may not be really translatable to humans also. So that's known, isn't it? Humans
Yeah.
may behave differently. So
Yeah.
there's a species difference between response to a particular antigen, Yeah, that's
Yeah.
right. So I'll take the last question here, which says that on one side, we are looking at the asymptomatic patients around 36%. I don't know how, anyways, and reinfection also taking place. So would you like to comment whether the vaccine will be useful or not?
Oh yeah, I think we definitely need the vaccines. So one if is that not every convalescent individual is having a
Protective.
antibody response, even for keeping the protective ability aside. In many individuals, we can't measure good antibody response after they recovered. So not everybody is mounting a response, right? And then two, that we are still understanding the potency of these neutralizing antibody responses, and we also don't know how much is there in the lung. So there are a lot of factors, and so I would definitely advocate for a vaccine, unless if it is proven that these vaccines don't work or they do any harm.
So asymptomatic, I think it has been discussed. Even Shiv mentioned there's a trained immunity in some people, which the innate immune response is trained enough to take care of the infection and there's no symptoms. So some people, they have that kind of immunity, already trained immunity, which you say maybe because of something BCG or whatever, there are so many things which are coming up at the
Yeah,
moment.
so that area is very interesting. It'll be interesting to see what people find. It's also interesting and confusing.
Yeah, there is a lot of mystery around. We are learning every day. So it has been great, 3 lectures. Thank you very much, Rama, for an overview of vaccine and what we are. So we have some panelists around. Dr. Amit Avasthi has put on his camera. Would you like to ask question, Amit? Anything to?
No,
Rama?
hi Rama, it's great to see you.
Hi, Amit.
It is always great to see you. I don't know why.
Same here.
What interaction we have is unbelievable. So I have no question. I know that you are doing really a great job, and we also talk offline about a few of the things. But I just want to congratulate you. That is a great talk, and Shiv also gave very nice talk. So this is great. Thank you.
All right.
Thanks for the opportunity.
Thank you, Rama, for sparing your time to give this very nice talk. And we all are educated. It was very informative. And everybody is waiting for a vaccine for SARS-CoV-2. And your lab is one of the top, pioneer labs where we are looking at something comes out. So we look forward to getting a very good vaccine from your lab. Thank you very much. And thanks all who have joined today. And we look forward to another session tomorrow morning. So join us back around 9:45 and we start another exciting day tomorrow. I close the meeting now. Thank you very much. Thanks all speakers. Thanks all panelists. And thanks Amit for waving hand. I see your smiling face. It's very good.
Oh,
I
you like
love
it?
you, man.
Great, thank you.
I love you. I love you. Yeah, yeah.
Okay.
There are few people you love. You know, this
I
is a
know,
guy.
I
You know, he generates so much of energy around.
I know.
So I like it. I
I
like it.
reciprocate the same.
Thank you.
Okay.
Thank you all. I
Okay.
would like to, I'm looking for a day that I can actually see everybody in person.
I know.
Absolutely. We are
Yeah.
actually dying to get back to the normal, but let's see how long.
So I
I
take
was ready to take my own vaccine and then start traveling.
very nice. Very nice. So I take this
FDA is going to hold
opportunity.
me back.
I take this opportunity to thank Dr. Kalpana Luthra also. I had to go for the meeting. I could not do that. Kalpana is around. So thank you, Kalpana, for chairing one session and handling everything. Thank you, everybody. God bless you all. Good night and have a good sleep. Rama, you have a good day. Bye-bye.
Thank you and good night.
Bye-bye.
Bye.